So thank you everyone for coming today. I just wanted to share some of the research that I'm doing out at the University of Queensland. I'm based at the School of Biomedical Sciences and the UQCCR, which is the Centre of Clinical Research out next door to the Royal Brisbane and Women's Hospital. So I'm currently funded by the Motor Neurone Disease Research Institute of Australia to do this research. So the title is Fueling Your Body to Fight MND. So what I really want to discuss is this issue of energy balance. It's not so much this typical thing that people associate with MND. A lot of people who look at MND research really, like Jake, they look at the genetics and they look at the cells and they look really deep down at really small mechanisms that might break down. And while I was initially trained to do this sort of research, I've taken a bit of a step back and looked at how the disease affects the body as a whole and to understand how it affects the body before moving back down to the small cellular systems. Okay, so we know that there is a lot of things that may cause MND. And this is primarily the reason for why it's so difficult for us to really understand what we can do to find the cure for MND. There are too many things, and a lot of people will refer to it as multifactorial. There are too many things going on at the one time to just target one system to cure the disease. You have to be able to target a lot of separate things that are going wrong to be able to make a difference to MND. So Jake has touched on the genetics. Rob briefly mentioned hyperexcitability, and what this really relates to is the activity of the cell. All cells send signals around the body from the brain that sends a signal down to the spinal cord, and they send signals out to the muscles to tell you how to move. And the problem is these cells in the brain, they have too much activity going on and they're just constantly sending signals, and the idea is if the cell is too active, it ends up just becoming toxic to the cell because there are no mechanisms or nothing to really control or dampen down this excess activity. There are these protein aggregates, which Jake also referred to in the genetics part of his talk, which are these proteins. All proteins in the body have some sort of function, and they're always folded and formed in a particular way so that they can perform their function. And when these proteins don't form properly, they tend to form these aggregates, and what's meant to happen in the cell is that these proteins that don't work properly are meant to be mopped up and degraded so that they don't sort of build up within the cell. And these protein aggregates are these proteins that haven't formed properly. They're forming these little globules within the cell, and the cell cannot remove them. And so lots of these misfolded proteins, they back up in the cell and they also become toxic. And that is believed to kill off the motor neurons. There's mitochondrial dysfunction. So what the mitochondria are, they're considered the little power plant of each cell. These guys, their jobs are to take sugars and glucose and fat that comes into the body and turn it into energy to be able to feed the cell. The cell will only survive when it has enough energy to survive. And the problem is in MND, what we see is a lot of these little powerhouses, they don't function properly, whether that's the ability to move the sugars or fat into the system to generate ATP, that starts to fail. And so these mitochondria, with more and more of them failing within each separate cell, you're going to get the death of the cells because there isn't enough energy to keep these cells alive. And then something that people have spoken about is really the environment. Is there anything in the environment that will trigger the disease? And it's really hard to know whether anything in the environment does specifically lead to the disease. There is a lot of anecdotal evidence of the toxins from the blue-green algae, exercise. A lot of athletes who develop MND, there's this idea that, well, maybe if you're a superior athlete that you're going to be predisposed to getting MND, but if that were the case, then every elite athlete would get MND. So the environment may have a role, and it's probably a trigger in the environment that sets the disease off in someone who would be predisposed to the disease. And that really comes down again to the genetics. So what I research is this idea of metabolic pressure in disease. So what we see is that in a lot of diseases, even in cancer or in Parkinson's, people tend to lose a lot of weight. And that's because the metabolic requirements on the body go up. And these people, they don't either consume enough energy to meet those requirements. So I think a lot of you would have learned maybe in primary school that energy in equals energy output plus growth. Yep. So what happens in a lot of diseases is the energy in does not really match what is required to give you growth. Okay. So the idea that I'm working on in MND is that as your disease progresses, or as the disease progresses, there's this increased need for energy to come in, because what you will see, or what has been reported, is that MND patients, they have this hypermetabolism, which means that at rest, their bodies are burning through more energy than what it should be compared to a normal person. So in a healthy population, you have this normal energy demand. You eat a lot of food. That's what we tend to do in today's society. And so what's in excess to what you need gets stored as energy. And so we tend to put on a bit of weight. And that's the excess food that we're consuming that is stored as energy. And when we need this energy, for example, if you were to go into a period of fasting, your body would depend on these energy stores, so the fat that is stored in the liver and around your waist, to basically feed your body so that you're not going to have some adverse health event. Now in MND, if you've got this hypermetabolism, so your body's burning through a lot of energy, but you've got decreased dietary intake, which a lot of MND patients tend to start having towards the later stages of disease. Mainly this is because they've got the weakness of the muscles, or they've got weakness in their hands, so feeding becomes somewhat difficult. So inherently, you have this decreased energy intake, even though that's not what you're really going out to try and do in this situation. And so you have reduced energy stores because you're not putting in more than what you need, and your body is already burning through what you've got. And so you end up having what I believe is this kind of cycle. And the reason why your body would have this hypermetabolism, I believe, is that the cells that are in the brain and the spinal cord and your muscle, they have this inability to be able to use the energy that you're putting into the body. And because your body is then trying to look for other sources of energy, you get this increase in metabolic demand, because it takes a lot of energy to burn through energy. Okay? And so you have this increased energy out. With malnutrition, you've got decreased energy in, and that naturally leads to your weight loss, because your body is starting to burn through your energy stores, which is your fat. And over time, if you have a depletion or a loss of these energy stores, you're going to have no more energy to feed the cells. And so the cells will die off because the energy demands of the cell aren't met. So why is this of importance to MND? And the idea is that if you're fatter, the idea is that if you've got a bit more body fat on you, that you're going to live a little longer than someone who has less fat. And this was a study that was conducted in 2011. And what they did was they basically looked at a cohort of MND patients, and they looked at their BMI, their body mass index, which we generally use as a measure of body fatness. And they found that people with very low BMIs tend to have worse survival outcome by this solid red line here. Those who have very high BMI, so if you're morbidly obese, you also tend to have a worse outcome. And this is probably associated with what people would consider metabolic dysfunction. So in someone who's very obese, their metabolic system is not as pristine as what it would be when they were someone of my BMI, for instance. So you know that when you're obese, you get things like diabetes. And that's because the whole metabolic system is changing to try and compensate for this. And in diabetes, you basically have a failure of your metabolic system. And so we think here in these morbidly obese patients, the metabolic system isn't functioning properly anyway, so they're not going to be able to use energy properly to sustain demand. Now, if you have a BMI between 25 to 35, you have improved survival outcome. So why would increased fat mass be important in MND? Of course, we all know that fat mass is going to be linked to food intake. And this study that was recently published this year in a very big journal 2014, this was a clinical trial study that they did with patients who were quite far progressed in disease. All of these patients were being peg-fed. And what they did was they supplemented the patient's diet. And they either supplemented them with nothing, that's the green line, the control, a high-fat, high-carbohydrate diet, or a high-calorie, high-carbohydrate diet. And what you can see is that in this trial that went on for over a year, you've got patients on the high-calorie, high-carbohydrate diet which have improved survival outcome. So something about the food that people are taking in, whether that's because it's specifically carbohydrates and calories, or whether it's because you're just putting more energy into the system to be available for the body, nobody really knows. And so this is what my research is really looking at. While dietary intervention seems to be a good way to go because there are positive outcomes from these studies, I understand that over time, dietary intervention may not be sustainable. And so the reason for doing my research is to understand why these diets and having a higher fat mass is of benefit. Because we don't want to keep telling patients that you need to eat 10 kilos of food even if you don't feel like it. We want to know the mechanism so that we can target that. So rather than eating 10 kilos of food, you may be able to take a pill that would target that system. Okay? So what we know about fat mass and nutrition versus survival in MND is that if you have a lower body mass index, you're going to be at greater risk of developing MND. If you have increased fat mass before you are diagnosed with MND, you tend to have less risk of mortality. Reduced fat mass in male patients throughout the disease will be associated with reduced survival. And that supplementation with the diet rich in calories helps stabilize your body weight. And so this increase in body weight might actually slow the progression of disease. So there's a lot of evidence coming out now, and this was really started to be looked at in 2010. And since 2010, there have been papers upon papers upon papers looking at this aspect of MND because it's something that is really important for the patients now. So it's something that can make a difference right now if we can figure out what is going on. So there's a lot of work being done in this, and we know that the association between fatness and MND drives us to understand why this is the case. So to do this, I use patients. Patients who volunteer at the Royal Brisbane and Women's Hospital, so I work closely with Rob Henderson, and we look at the patients. And I also use a lot of mouse models because I'm trained as a basic scientist, and all of my research growing up until now was done in mouse models. So I'm very good with my mice. Okay. So what we wanted to do, first of all, was to look at the population of patients that we have. If fat mass is so important in MND, what we need to understand first and foremost is that all of the physiological mechanisms in the body that tell us about fat mass is not affected by the disease. Okay, so there are certain hormones and proteins that are secreted from the fat, like leptin and adiponectin. These are secreted from the fat cells themselves, and they give you an indication. They're markers in circulation that you can measure in the blood and plasma, and they tell you how much fat mass a patient has. So in normal people, like healthy people, leptin, it increases. The fatter you are, the more leptin you have in circulation. Whereas adiponectin, the more fat you have, the less adiponectin you have. Now we all know about insulin. Insulin is one of these hormones and proteins that are involved in taking fat out of the system because elevated fat in your blood is not good. What that does is it takes all the fats out of circulation and puts it into storage. So insulin really is what makes you put your fat around your waist. And then C-peptide, it's a protein that's secreted with insulin at the same time, and it actually helps bind the two parts of insulin together to make it a functional protein. Now, when fat mass goes up, insulin and C-peptide also go up. This is what happens in the normal population. In our MND patients, we can see that all these systems are intact. There is nothing wrong with them. In our patients who have higher body mass index, they have high levels of leptin. Patients who have higher body mass index they have lower amounts of adiponectin. So these lines, what people like to call them is this correlation analysis. It's looking at the relationship between one thing versus another. So if you plot your body mass index against the amount of a protein or hormone in circulation, you can draw a line to see how good that relationship is. So that's what these lines here represent. And then here with insulin and C-peptide, as fat mass goes up, so does insulin and C-peptide in our MND patients. Now, moving on from this, what I've been using the plasma from the patients for is to really screen for anything that might be different between MND patients and normal people that might tell me something about their metabolism. Because they're supposed to have increased metabolism, is there something that we can detect in circulation that will give us a good indicator of this? Because taking a blood test would be relatively simple. And so with leptin going down, and we see it going down in patients who have longer disease duration, because of course you're losing fat mass, we took this cohort of patients and we went and looked at this factor called GIP, and of course insulin. Now GIP is gastric inhibitory polypeptide. All these proteins have these crazy names that I'm still learning. But really, regardless of the name, it has a certain function. And that is, it's supposed to make fatty acids. So it's supposed to make fats in the system. And it's supposed to help insulin take the fats from circulation and put it into storage. So what we see in the MND patients is that this peptide is decreased in the patients compared to our control population. And this occurs regardless of the level of insulin. The insulin is the same in our patients. So you would think that insulin functions normally, although we have no evidence to show that in our patients as yet. And there are many tests that we can do to show that insulin is working properly, but these can be quite arduous. So it's a matter of getting patients into the clinic to be able to conduct these tests. But what this decrease in GIP could mean, or could underlie or cause, is what we see here, is that the rate of body mass index predicts prognosis. If you have a decrease in the expression of the peptide that is supposed to make fat, you would presumably have less fat. And so with patients over time losing fat, that would seem to fit the whole story. What this also might fit is this observation a few years ago that patients who have high triglycerides, now triglycerides are the fats that are in your blood, and sometimes they're meant to be bad for you because they cause heart disease. Patients who have elevated triglycerides and cholesterol have better survival. So this decrease in GIP might be causing for insulin to leave these triglycerides in circulation as a means to compensate for the disease process. Okay. So to really understand what's going on, because it's very difficult to work in patients, it's hard to get a good control population. How do you get someone who's 70 years old, who has the same body mass index as an MND patient who's 70 years old, because they could be quite different? How do you get a 70-year-old who's not on cholesterol drugs or statins or anything to kind of help their general health and well-being? It's very hard to find a control population. So I look at the mouse model. And this is the mouse model that Rob was talking about earlier today. It was the first mouse model used for motor neuron disease. And this guy, Gurney, made his career off this mouse. He developed this mouse. It develops MND symptoms. Okay, so the problem is with SOD, and Jake saying that the SOD mutation only really accounts for a small amount of MND cases. To date, this is the best mouse model that we've had to use to be able to understand MND. With this advance in genetics and the advances in the ability to generate mouse models very, very quickly, we would hope that we could get a C9orf72 mouse model very soon. And my goal would be to show that everything that we see in the SOD mouse would be conserved across all different mouse models of MND, because I believe that this metabolic component is common to any type of MND that you will get, because people do lose weight. Okay, so we use the mouse at different stages of disease. We've characterized the mouse so we can study it at very specific stages. It either has no symptoms, which is a benefit of using the mouse model because finding MND patients with no symptoms is almost impossible. We use mice at the onset of disease. This is when they start to develop weakness in their hind limbs and they get this tremor. And then at the end stage of disease. This isn't the end stage of disease because ethically we do not allow our animals to progress to the end stage. These animals have severe paralysis and we sacrifice them for ethical reasons. We don't leave them to the point where they can't eat and they can't drink and that they're being taken over by the disease. Okay, so a lot of our studies are done in these mice. So we know in this mouse model that they lose motor neurons. Very similar to the pathology we see in patients. As the animals get sicker and sicker, the number of motor neurons, which are the cells in the spinal cord, these ones project out to the muscle and they basically relay the information from the brain out to the muscle. So these guys are a really important midpoint for the sending of information around the body. Now these guys, you can see that in the wild type, which is a mouse that doesn't have the mutation for motor neuron disease, you will see that there's quite a few of these really big cells in the spinal cord. In the SOD mouse, the MND mouse, there's only really one. So we have a very specific way that we look at these cells, and these cells, to be considered a healthy live cell, have to meet specific criteria. And so as these mice get more weak, we see that the number of these motor neurons become less and less. What we also see, and I'm not sure if you can see the image very well down here, this is called the neuromuscular junction. This is where the cell in the spinal cord makes its final connection with the skeletal muscle. And what happens is the nerve from the cell, it starts to send out a projection to the muscle. And if you can see it, it's labeled in green here. This is the nerve. Now, at the muscle end, you have these proteins that respond to the chemicals that are being released from the nerve. And these proteins are labeled here in red. Now, what you will see in a healthy connection between the nerve and the muscle in a normal person or in any normal, you would still have normal neuromuscular junctions in MND, you would see that the nerve and the proteins in the muscle, they sit directly opposite each other. Because once the chemicals are released from the nerve, you have to have that response, immediate response, to have muscle contraction. If the chemical has to travel a certain distance, you're going to get delayed muscle contraction. So the way the neuromotor system is developed is so that these two structures lie directly on top of each other to be able to induce muscle function. And what you will see in MND and even in normal aging, when you sort of start to slow down in your movement, is that these nerves start to pull away from these proteins in the muscle. And so you get less muscle contraction. And as we see this in MND patients, we also see this in our mice as the disease develops. What we also see is that these are the standard behavioral tests that you do in a mouse to show that its muscles aren't functioning as well as what they should be. Here, the wild type, the normal mouse, is denoted by the red spots, and the MND mouse is the blue. And you can see that the rotarod, which is just this little machine, it's a rod that turns around in circles, it just rotates, and you put the mouse on it and the mouse just runs. And you can see how well the mouse performs in that test. And this test isn't necessarily about the strength of the muscle, but more the coordination and how the signals are being sent around. And you can see that over time, as the animals get older and they're developing the disease, their performance on this apparatus becomes worse. In the mice, the main thing that we look at is their grip strength, because a lot of the mice use the hind limbs for their strength. We test the grip strength, so we put them on a force transducer and we just give them a little yank back, and we can see how much force they have in their hind limbs to grip onto that apparatus. And you can see that this is where our mice develop the onset of symptoms at 70 days of age, that there's a decrease in that grip strength. And as their disease gets worse, you can see that the grip strength starts to get worse as well. So what we also know in the mice is that they lose weight. So after they develop the symptoms, and they're eating food, they're still running around, they're able to do what they can do. They're not at that point where they're just sitting in the corner because they're so paralyzed. We never let our mice get that way. These mice tend to be very, very good at hiding pain or any problems, because I think that's an evolutionary thing, the survival of the fittest. So when you look at a mouse, it's very hard to tell that it might be sick. But we can see that when we scan them that they're losing a lot of body weight once they develop those symptoms, even though they still look fairly normal. And we can see that a lot of this loss of body weight is fat mass. So even though they lose weight from 20 weeks, they start to lose fat at 19. So that loss of fat mass is preceding their total weight loss, okay? And what's interesting is if you feed these mice a really high fat diet, just like we've done in the patients, you can extend the survival of these mice. It's because of these studies in mice that people have gone on to put high fat diets into the clinical situation to see what will happen. And what's really good is what we've seen in the mice here has translated into the clinic, where patients being fed high fat and high calories will have improved outcome. So what is the mechanism that's causing this? Why is fat so important? Why is fat, and how is fat improving survival? So what we've gone on to do is we've looked at the fat mass of the mice. So in the mice, this fat mass here is called the white fat mass. It's inside the body here. And when you open up the mouse, you'll see really nice big pieces of white fat. Now this white fat really equates to the fats that we use as an energy store around our hips, our bum, that's all those kinds of places that we don't want the fat to be. That's the fat that we're looking at the equivalent of in the mouse. And what I've done is I've looked at the fat in mice that have no symptoms, mice that are starting to develop symptoms, mice that are at the late stage of disease, and mice I've put in this new stage of disease called the mid stage, because from the onset to the end stage, there is a lot going on. And I was concerned that I was missing something between that point. So I've added another time point that sits within the middle of these two stages. And I've analysed mice at this age as well. So what these little dots show is basically a gene analysis of they're genes that encode proteins, so they get turned into proteins, and these proteins are involved in breaking down fat and moving it out of storage for use as energy. So I've done a screen for these genes to see if anything might be different. We've got, in the graphs that you see, these are called volcano plots. The line in the middle represents 0. Anything to the right of it is an increase in expression. Anything to the left of that is a decrease in expression. So what we can see in these mice is as the disease progresses, they have an increased expression of genes that are involved in breaking down and moving the fat out of storage. Now the problem is with analysing genes, that's just the message. You don't have a functional protein. The proteins are what do all the work, so the genes don't necessarily mean anything. So with this in mind, with this data in mind, and this is a bit of a heat map of these genes, you can see that all these that are involved in breaking down fat and moving fat out of storage, and this thing called ketogenesis. I'm not sure if many of you have heard of people trying to use a ketone diet in motor neuron disease. So ketones, what they are the final source of energy. What happens is fat, your body depends on things like glucose, your sugars, then it starts to depend on fat. When your body becomes so starved to the point where it doesn't really know what to do anymore, your fats get changed into ketones, because these are the energy substrates that are used by the brain to keep us alive. So the ketones are the last resource of energy. And we can see that these mice have an increase in genes that are involved in this pathway. So does this mean anything functionally? Can we measure a change in the fat mass rather than just weight in our animals? And what we've done is we've gone back to the mouse and we've taken fat out of these mice at the 4 different ages. And what we've done is we've just incubated them in a physiological solution. And over time, what fat does normally is it releases fat out into the system to be used as energy anyway. And we thought if we could measure this release of fat, could we see an increase in the MND mice compared to the normal mouse that's not sick? And we can see that just with these, over time, we've taken time points at 30, 60, 90, and 120 minutes of the little fat bubbling away in circulation. You can see that here it seems like there's an increase. Right at the end of the disease, when the mice here have a lot of difficulty eating because they've got hind-limb weakness, so they can't reach their hopper as well. And we put food on the floor to compensate for that. But it seems at some stage that these mice may not be eating as well as what they would because they're developing this weakness anyway. And it would seem that at this stage, when they're not putting in enough energy through their diet, that they may be increasing the amount of fat that's being released from these fat stores. But if you plot these line graphs against each other, what you will see is not necessarily that it's an increase, it's just that the amount of fat that's being released stays the same. From a very young mouse through to a mouse that's sick, it doesn't get this decrease in fat release. This decrease that we see here is very, very normal because as we get older, we get fatter because the processes in our body that drive the release of fat start to go down. So what we see in the wild type is normal, and what's happening in the MND mice is that they don't have this system in effect. They're actually just maintaining the release of fat in their system. So where is the fat going? The fat is obviously being used for something, but we don't know what it's being used for. And the first thing we wanted to look at is this concept called non-shivering thermogenesis. So energy that is not used by the mouse in any physiological system is lost as heat. And there's this type of fat in the mouse called brown adipose tissue, it's BAT, and this is what you see in babies. Babies have a lot of brown adipose because they stay warm by non-shivering thermogenesis. So mice also have this large store of brown fat on the back of their shoulders. And what we did with people in the US is we measured the amount of glucose that's being taken up into this tissue. So what you can do is you can inject a radioactively labeled form of glucose, and any energy that's required in the form of glucose by any organ, it will basically take the glucose up to use it. And what we've done here is we've really looked at how much glucose is moving into the brown fat to see whether it's because the brown fat is more active, and so the animals are losing that energy as heat. So these are basically the cross sections through the mouse this way. Okay, so, and then this is cut through the abdomen this way, and this is from the top view. And you can see that between the wild type and the MND mouse, the amount of glucose that's going into that brown adipose is the same. So to us, this is a very important experiment to tell us that the mouse isn't losing the energy, it is using it for something. So I next wondered what it could be. And I decided to target the skeletal muscle because we know that the skeletal muscle forms a lot of the human body and it's very energy demanding. So glucose is used as an energy source in the muscle. And what I decided to do, similar to the fat, is look at the expression of genes that are involved in breaking down glucose into energy. And like I said before, to the left of the line is decreased expression. So what we can see is that as the mice get sicker, they've got a decrease, in the expression of genes that are using that glucose for energy. Okay. So this is the heat map once again, and the heat map, the decreases that we see, the fold change, so the amount of difference that we see compared to the normal mouse, it's going, I'm using the colors as a measure of how big the change is. You can remember or appreciate that the changes for the fat were somewhere in the yellow-orange, so it was a small increase. What we're seeing here in the glucose genes is going down to the black area, so there are really, really big changes going on in the skeletal muscle. So would that mean that the skeletal muscle in these mice is not able to use energy? The systems that are in place for breaking down the sugars that come into the body through your diet are not working, and so the muscle is not getting the energy that it needs. And so would this mean that the fat that is being lost is moving to the muscle to try and basically feed the muscle to maintain function and survival? And what we did was we took out some muscle and we had a look at the amount of fat that's in the muscle. And we can see that this mid-stage, after the mice have developed symptoms, when they start losing that fat mass and losing weight, is when we see this increase in the amount of fat that's in the skeletal muscle. So for us, it seems that yes, the fat is moving to the muscle to be used. And with some researchers in Strasbourg, we've shown that this change in fat can really be important when it comes to an experiment where you look at how mice perform in different exercise regimes. So if you take an MND mouse and you run it on a treadmill at really high speeds where it's just the speed keeps increasing, so you're basically putting the mouse through high intensity exercise, what you would equate that to is the use of glucose. So when athletes run, the 100-meter sprint, the muscle uses all of the energy that's stored in the muscle, and that is the glucose. When you're doing long distance running and you're going for, like Trent does, like marathons, I don't know why he does them, but you would see that the fuel that's being used in your muscle changes to fat if you're doing sort of a gradual, moderate exercise regime. And we've seen in our mice that our MND mice perform better on a moderate exercise when their muscles are using fat than when their muscles are using glucose. Yeah. And what we also see is this increased expression of ketones, which are those substrates that are used by the brain. So we think that what is happening is as fat is moving out into the system, it's being broken down to feed the brain, possibly the spinal cord. I have an honours student who's working on that aspect at the moment, and of course the skeletal muscle. So what we wanted to do then was we decided to look at growth hormone. So everyone knows that growth hormone is an anabolic steroid. People pump it into themselves to build up their muscle. So the idea is if you're losing the ability for muscle to work properly, would giving it growth hormone help the muscle to function? There have been a few clinical trials earlier, 2000s, 1990s, that have looked at growth hormone treatment. And growth hormone, unfortunately, does not do anything for MND patients. And I believe I know the reason why, because of the experiments that I've done. So, my picture got lost. But what happens is growth hormone works in balance with insulin. So when growth hormone levels are very high, insulin levels are very low. So that means that with insulin being low, and insulin is supposed to pull fat out of circulation and put it into storage, you're going to get very low levels of fat mass. And that with low insulin and high growth hormone, you actually get the movement of fat out into storage, out of storage. When you have high levels of insulin and low levels of growth hormone, what you will see is fat will just accumulate. So we thought there's an inverse relationship here. As growth hormone goes up, as insulin goes up, growth hormone is down. So maybe if insulin is so important along with growth hormone with regulating fat mass, what happens if we develop a mouse that cannot use its fat? Okay. So we generated this mouse here. This was the first mouse of its type in the world. It's an MND mouse that has no growth hormone signaling. And of course, with no growth hormone, it's also very short. So this is an adult mouse, and it's about 6 centimeters long. So they're really cute, but really cute, really cool. And what happens in these animals is metabolically, they become very, very pristine. They're very insulin sensitive. Now, insulin is the hormone in your body that tells your body to use glucose as the energy source. It facilitates the breakdown of glucose for energy. And so we wanted to know, well, what happens here? These animals, growth hormone deficient animals, also get very, very fat, and they're also resistant to cancer and a lot of other diseases. So we thought maybe it would have two effects. It could improve the survival of the mice because it's not going to develop metabolic disease or cancer or anything like that. Or it could have the opposite effect with not being able to use fat being the critical energy source, that we may see a worse outcome. What we see is that this mouse that has no growth hormone signaling, we've got 3 different mice here. This is a normal SOD mouse, the standard SOD mouse that we've been using, the MND mouse. This is a SOD mouse that has half of the growth hormone signaling. The way we generate these animals is you'll always have one that has no growth hormone signaling and one that has half. And for me, it was very important to always look at the midpoint. You can't just go for the 2 extremes. So we also generated these mice that have half the amount of signaling. And this is an MND mouse which has no growth hormone signaling. And this is the survival curve, the typical survival curve you see. And the SOD mouse and the SOD with half the amount of growth hormone signaling and the mouse with no growth hormone signaling, they live the same period of time. It doesn't extend survival and it doesn't exacerbate and shorten lifespan in these mice. What it does do, though, is it delays the onset. In our mice that have no growth hormone signaling, the disease onset of the symptoms, the muscle weakness, is delayed by 2 months. So they're not developing weakness until much later on in the disease when compared to an MND mouse. But the problem is, as soon as they get the disease, the progression is very, very speedy. And I think the reason why there's this delay in onset and this very rapid progression at the end is because at the very beginning, the mice are able to use glucose very efficiently, which is the energy source for neurons, and it's the energy source for the muscle. But once the animal develops the disease and it cannot use that alternate energy source, the fat and the ketones, there is nothing left that is available to maintain the survival. And I think this is why these animals tend to have a very rapid course of disease at the end. So in summary, I really believe in my research, first of all, which is important. And I think that the metabolic homeostasis in MND is a really important aspect that we need to look at, because if we can change this now and we can understand how this helps improve survival outcome through increased fat mass and increased nutrition, can we modify the course of disease for MND patients right now? Can we extend survival? But if we cannot extend survival, can we improve the quality of life by delaying the symptoms and the rate at which they progress? And I believe that by understanding at the whole body level how these changes in metabolism affect the person, we can then take things from the big picture down into the cell. Because just like the whole body, a cell needs energy to fuel survival. And if the same mechanisms that fuel the energy for survival are the same as in the cell, we can start to target those mechanisms and hopefully save the cells as well. So what I'd like to finish off on is a quote from a physiologist. He was considered to be one of the best scientists in the world well, I think he still is, but a long time ago there was this quote. And really what it comes down to is all of the mechanisms in our body, they're all varied. This is the multifactorial nature of MND. They have one goal, and that is to maintain the balance inside the body for survival. And it's all of these mechanisms, not just the one, it's all the vital mechanisms that we need to consider if we're going to have a world free from MND. Okay. So with that, I'd just like to finish everyone who's involved in the work and all the supporters, MND and Me Foundation, of course. Thank you. I think you should eat whatever you want. And no, I think you're free to eat whatever you want. Maybe even coconut ice cream, because coconut, it's one of these medium chain fats. So fats, there's your short fats, your long fats, and your medium fats. And what happens is when fat gets put into those powerhouses, those mitochondria, to be broken down for energy, it goes through multiple steps. Each time it goes to the step, you break a little bit of the fat off and you break a little bit of the fat off. And so for the body to break down long fats also takes a lot of energy. But the interesting thing about the medium chain fats is they go straight into those powerhouses. They don't need to be transported across. And that's why a lot of people are trying to look at medium chain oils, coconut oils, as a treatment for MND. So coconut is good. Well, in normal people, in the general population, they're not normal. In the general population, too much coconut fats and oils are associated with heart disease and whatnot. But I think in this situation, when the energy is being used immediately, that it's not going to be an issue. You don't have to eat salad. I think you should eat pies and ice cream and everything. You've got to eat more. That's that hypermetabolism, that increased resting energy of burning more than what your body should be doing when you consider your BMI, your age, and your gender to someone who's equivalent. That's right. Yeah. So this hypermetabolism was really discovered a few years ago, mainly by the French. So Charcot, who Rob alluded to right at the very beginning, he was a Frenchman, and he was the one who sort of first described the pathology of MND. And the groups who are really leading the field in this metabolism and who we work with are in Strasbourg, and we've done that experiment with the mice and the different exercise regimes. So yeah. You must eat more. Yes.
And it's very difficult when you exercise and eating is hard. It takes time and you just don't have that.
That's right.
But what's a quick fix to this? Is there any something?
I would like to find a quick fix.
No,
but,
the energy, can you just get something and
no. You're just going to have to eat at the moment. Unfortunately, copious amounts of food. But you just change your relationship with food, seeing it as something good. Because I know we live in a society where we tell people don't eat too much. This is the opposite. I don't care about normal society right now. We're going for enjoying food and just eating it. So, yeah.
there's also a risk of high potassium.
Yes.
How much is enough? How much should you have for eliminating the risk?
I believe that. Oh yeah, sorry. The high amount of coconut that you take in your diet is also associated with increased potassium. Okay, and that is not good for the body. We know that. So it's really hard to know what is the right level of coconut oil. Like, it's hard to know. When I said people are feeding high fat, high carbohydrate diets, is it the fat? Is it the carbohydrates, or is it just energy? And so we don't really know the contribution of each of these dietary substrates to the course of disease and whether it's going to improve disease or not. So we've recently teamed up with someone in the Netherlands. He oversees the ALS Centre of the Netherlands, and he's done a lot of studies on dietary intake as a risk for developing MND. And so we're teeing up with him, and hopefully we're starting a research project next year where we look at the different components and how much coconut oil has this person taken and how does that correlate to severity. Or even water, it's the energy, the fat, it's the chain of the fat. And along with looking at coconut oil, we're also looking at the vitamins, minerals, amount of fat, amount of glucose, amount of protein, carbohydrates that are in your diet. And the idea is not to restrict patients to a specific dietary regime, because I think that's really hard to control. The idea is to understand what patients are eating and then correlate that to their outcomes, their disease progression, their clinical features, and to see how that correlates and how that really works out. It should be able to tell us a lot about someone's diet.
And the amount of protein in the diet affect the progression of the MND?
Having too much protein, I don't think would affect the progression of MND. There's been 1 or 2 studies looking at the amount of protein that patients on a high protein diet, they don't have improved outcome, but their outcome isn't worsened either. It's just the same as what it would be if they were just eating normally. So the high protein doesn't seem to affect the course of disease. I mean, for someone who's not afflicted with the disease, high protein can be bad, not because of just the bowel cancer, but it changes your insulin sensitivity. So it really messes around with your metabolism.
What about preservatives?
Preservatives. Okay.
Things around tea, boxes and stuff. I'm very like, I look at mine, just have a coffee.
It's a bit of anxiety there. Yeah, so the problem is a lot of people haven't studied diet, because even though it was described in the 1990s that we know for a fact that MND patients do not consume enough nutrients, no one's really taken the initiative to look at the diet because they just consider it to be a normal part of the disease. People are normally just going to eat less. So why would looking at the disease make a difference when you look at their diet? And so it's only after this whole body mass index thing that people are really starting to look at the diet. And is it particular things that we're eating that might be triggering something or predisposing you? And that's the person in the Netherlands who we're working with, because he's been very good at looking at that sort of research.